One thing that I see in common with the people that are up here talking is an unbelievable amount of passion. If you think about the work that's being done and the work that needs to be done, it's monumental. And it's being done by people who are truly passionate. And I hope you get a little bit of that when you see some of this work. I get to represent a team. I do pediatric surgery and fetal surgery about half the time. The other half the time I run my lab at Mayo. And I today get to represent the work from that team. So we'll start off with the work as a fetal surgeon and pediatric surgeon. And we like to say at our center, and if you ever meet the rest of the team, you'll get the same flavor from them. It's about meeting an unmet need. And I think this will help you understand that a little bit. So this is our unofficial goal, keeping the tiny humans alive. So this is why we got started in this work. This is the Casellas family. So this is a family like almost every other family in America, very middle income family. They've got jobs, kids, and then a problem strikes. And this particular family had a problem. They had a twin pregnancy where just a few years ago, both twins had a better than 90% chance of dying. And in that group of, and we're going to talk a little bit about this, but in this group of disorders or problems, it's called twin-to-twin transfusion syndrome, where basically one twin is connected to a placenta that's also the same placenta as the other twin, but they have two separate sacs. And this happens in about a few percentage of all twin pregnancies. Twin pregnancies are fairly common, and they're getting actually increasingly common, about 1 in 100 to 1 in 90 pregnancies. In twin-to-twin transfusion syndrome, you've got an imbalance, and we're just going to talk about a few diseases now. You've got an imbalance of the blood share from the placenta where one twin is donating blood and the other twin is receiving that blood. Too little, too much. Pretty simple. And what happens is the donor twin goes into renal failure. They don't have any urine in their bladder, and they begin to really suffer, and their demise is almost imminent. And the other twin has too much volume, too much blood, and their heart goes into strain and failure, and they too are in an imminent demise situation. So this family is now, after 20 weeks of gestation, been told this problem, and they're told now, at a time when maybe only 1 or 2 centers around the world can do any intervention, basically you've got to get in your car, drive to the airport today, go to this center, because if you don't have a procedure done, which may be 1,000 miles away, or at one point was just on the other side of the globe, your babies are going to die, and there's nothing else we can really do about it. Originally, we used to take a small needle and just drain off some of the fluid, and it was a temporizing measure. Modest improvement from 90% double demise, where both babies died, to where we had 60 or 70%. So we only have a modest improvement. A group of collaborators out of Belgium, Jan de Press, published a paper that looked at being able to sever those connections between the two twins in a procedure called fetoscopy, or laser fetoscopy. And what we saw when we did this is that you had an 80% reduction in the neurologic morbidity and twice as likely to survive. And this was in 2004 when we were just beginning it. So we took a disease that had a survival rate of only about 10% and we improved it to make it 50 to 60%. And today at our center, it's about 75% of the twins now, both twins will go home, and 93, 94%, at least one twin will go home. That's a major improvement just in just over a decade. This procedure itself is interesting because it's all done through very minimally invasive techniques where you gain access to the uterus through mom's abdominal wall through a small little incision and use a camera with a laser fiber at the end of it to be able to visualize these vascular connections. And when you see these vascular connections, the goal is and you've got to identify them properly. You don't want to take too close to the donor, too close to the recipient. You want to be able to take them right at the vascular equator, and you want to be able to disrupt them. When that happens, and I'll show you a video what this looks like, when that happens, you disrupt that imbalance, and that regular flow from one twin to the other twin stops, and they can catch up, and that takes just a few days to a couple weeks to see. But this is what we see when we go through that small little port through mom, and the lighting isn't great here, but you can see the baby And these are the pictures that we send to mom right after the procedure. There's the mouth, the nose, and this is an 18-week gestation. So this is well before viability of a baby. And these are these important vascular connections that we're mapping out to find the vascular equator. And once we figure out the vascular equator, then the next goal is to take a small laser fiber in this environment, and this is magnified. We're only about a centimeter away. Too far away, you don't get any heat transfer, and too close, you'll rupture that vessel, and it's going to be a demise for both babies. And what's interesting about this is the uterus sits on the mom's aorta, and it's moving 80 times a minute, right? And you've got to stay a centimeter away. And the baby's heart rate is about 150 per minute, and your heart rate, because of those two things, are about 300 per minute. So this is what you get at the end of it. You get a placenta. This is an actual placenta that's been what we call dichorionized, and basically through the laser fiber cut into two. And this is what they really get. Two babies that are born healthy and get to go home. And this is our mascot, our model for our center. So being able to see things like this, you think about the work that we do in any field of medicine, any field of research, it's all important. And I can't value one person's work or one area of work more than others. But when you talk about fetal intervention, you're talking about contributing 80 years of life and productivity. And I think that when you can couple that with a reduction in morbidity, mortality, and cost, improving outcomes, improving the system-based operations within a hospital, that's pretty special. And this is the family as they grow older. They forget about you, actually. After a few years, they stop sending you these pictures because they're doing so well, and that's great. They don't need you anymore. And this leads to some of the most exciting things that we do is open fetal surgery, where basically you're doing an operation that can't be done through small little incisions through the abdominal wall, but rather big operations that require opening up mom, exposing uterus, opening up the uterus. And if you can see here, this is the last surgery I did with my mentor in Philadelphia at CHOP. And this is a little foot poking through here, and we're going to operate on the back of this baby, repairing the spina bifida. So spina bifida was a problem that is a problem that's devastating. These kids, the morbidity and even the mortality is really substantial, and for 50 years we haven't really made much headway in this disease. And it's interesting because spina bifida, was really the first major open fetal indication that we came up with in medicine that allowed us to push the frontier of open fetal surgery. But we needed an indication that you could prove a need as well as a safety profile that you could take through a clinical trial, like a legitimate clinical trial. My mentor, Scott Adzik, and his team produced this first body of work in 2011 that showed that kids who got their spines repaired in utero, so about halfway through gestation, had a significantly better quality of life. They were walking much more often, 50% of the time, than kids who weren't, who then are in wheelchairs. They didn't need shunts. Their shunt rate went down. These shunts where you have this fluid in the brain that have to be delivered elsewhere and all the complications. Neurocognitively, these kids were the same as their peers and their siblings. They didn't have that neurocognitive decline that's severe. And the general cost, one of these kids with a fetal repair versus a postnatal repair is roughly 10% the cost to the healthcare system. It's a pretty dramatic impact across the board for the patient, for the family, and for the system. So spina bifida, just briefly here, and I'm going to test everybody when we're done. This is a normal baby. This is a brain, cerebellum, spine here. Spina bifida is at any point in the back where you've got an incomplete covering of the neural elements here that are exposed to the amniotic fluid. And this is the most severe form of spina bifida, where the neural elements are exposed to this amniotic fluid that is really just toxic to the neural elements. And it's a progressive neurocognitive decline that you see here in the brain. It's progressive injury to the nerves here that allow for injury to the sensory and motor neurons from the level of the defect down, as well as problems with the hindbrain slipping into the bony portion of the neck because fluid is not only leaking in but is also leaking out. So the goal of the procedure, and we'll talk a little bit more about this, and I'm going to speed this up. The goal of the procedure is to expose the back of the baby here, and while opening up mom, uterus, and the back of the baby here. And you can see the spinal cord perfectly in view, and it's just free-floating in the amniotic space. And the goal is to be able to close it up just very similar, almost exactly the way we do postnatally, except this is hard. Nothing about the baby's physiology, nothing about all the advancements we've made in the last 200 years with anesthetic and medicine prepared us to be able to do this, because almost everything you want to do for mom from a general anesthetic standpoint is the exact opposite of what baby needs. Every little thing you do to make it a little safer and a little better for mom makes it a little more unsafe and a little more difficult for baby. So this is something that had to be developed over a great deal of time, and it's something, if you look at from like a transplant center standpoint in the United States, there's over 300 abdominal organ transplant centers in the United States. There are fewer than 10 advanced open fetal surgery centers in North America. So this takes a great deal of effort and expertise, not just from the surgery perspective, but the whole team. The amount of people that are required to be able to do this procedure is really substantial. I'll give you guys a little bit of an idea what this looks like. So, and no one's squeamish, right? So we've got the uterus here exposed. And then we've mapped out where the placental margin is, where I need to be far enough away from the placenta so we don't have any problems with that. And we use ultrasound to help us guide us inside the uterus. Then we place these stay sutures here, and we use a special stapler that's only made for this indication. They're Vicryl staples that reabsorb. So we know that if you use metal staplers, it's actually like an IUD, the mom can't get pregnant again. So we had to come up and develop technology or equipment, and this seems minor, but this was pivotal in this operation. Secure the membranes, the blood supply to the uterus is amazing. It creates what all the surgeons in the room know, audible bleeding if you get into it, which is bad. So from our perspective, this was a big part of an operation and a game changer for us. We mobilized the baby's back into view, so now you've got the baby sitting there with this is the spine, this is the neuroelements in the spinal cord exposed, and this is the defect here. And we're going to close this in multiple layers. And again, you're operating on a fetus, so you've got to hurry this along. The tissue is not great. And then at the end of that, we're going to close up mom's uterus and watertight and place that all back in, hoping that we get another 12 to 13 weeks of gestation. And often we do, and this is the first one that we did here in Minnesota. And this is little baby Claire. They've given us permission to talk about them. But this baby here in this family, just to recapitulate some of the data, that this family now has a child that will likely walk when she wouldn't, based on the defect. This was a high lumbar defect, so should be paralyzed from here down. Now this baby today is 3.5 years old, and she can walk, she can stand, she needs some orthotics and assistance. She's never needed a shunt. She has the same IQ, neurocognitive function. Her Bayley's 2 score is the same as her siblings. She has just a fraction of the problems that her counterparts who undergo postnatal spina bifida repair have. She's a remarkable case for us, and we've done over 30 of these here just in our center in Minnesota. And then you get great pictures like this where this is a 1-year-old picture. This girl walks completely unassisted, never needed a shunt, and now she's 3, and her Bayley 2 scores from a neurocognitive standpoint are the same as her siblings. And this is her 1-year-old picture. Her bear has the same operation scar as she does here. So then you can do really rare things. Tumors that used to be fatal and terminal in the 20 to 30 week gestation period for families. This is a very rare tumor called a sacrococcygeal teratoma. Generally speaking, when they were giant, this size, it almost always led to a demise. Well, now we're the second center in the world that's been able to do this, where we've been able to resect or debulk the tumor and allow for the heart, which is the real organ that's stressed in this situation from a vascular steel standpoint, to recover, and for the babies to make it later into gestation. You can see this is the massive tumor. And this baby weighed 500 grams. The tumor weighed 640 grams, so bigger than the baby. It's quite remarkable. The goal is not to take the entire tumor out, but to debulk it here. And once done, you can see that there's a tiny little IV here that's placed. It's critical. You can't do this case. Here's the placenta. You should never be able to see that without delivering it. And then at the end of the case, here's where we're taking off the tumor, debulking the extra corporeal part of the tumor, and then we're closing up the wound. You can see here that both legs are now in view, and this is where the tumor used to lie here. This baby went on and was delivered 7 weeks later and is now totally healthy, tumor-free at a 1.5 years of life now. So fetal surgery is an important part of what I do, but really it's about an idea of treating diseases very early in their process. And when we think about a lot of the treatment strategies that we have in medicine, it really is less geared towards preventive medicine, which you've heard a lot of today, that smart thinking about trying to anticipate problems, anticipate need, to more curative, anticipatory type approaches where you say, well, listen, take diseases like inborn errors of metabolism of the liver, where you've got a single gene disorder, vast majority of these issues, single gene, right? It'd be like having, the White House. And I hate to bring up the White House right now. But it's like taking the White House, this massive building, and if the doorknob is broke, right? Tearing down the whole White House, right? The whole building, and rebuilding the whole thing just because the doorknob is broke. That's what we do today. That's how gross, and I really mean that, that's how gross we are at the level of medicine that we're at right now, because most of these patients require a whole organ transplantation or some supportive therapy that really doesn't address their problem, when all they really need is the doorknob changed. And that's the genetic approach that I want to tell you about next. Our commitment from a fetal care standpoint is to not only advance the clinical practice that is immediately available to patients, but to advance what the clinical practice will look like tomorrow. So we've chosen this group of disorders at my lab. We're interested in gene therapy, and that really means ex vivo gene therapy, in utero gene therapy, or in vivo gene therapy. And then we spend the majority of our time on is gene editing, gene correction, which is probably the second or third iteration of gene therapy as it comes out. So I want to talk to you guys a bit about something that is just around the corner. Our FDA packet is in. Others have submitted similar type packets to use similar type of technology for their gene therapy approach. But I think this is helpful for you guys when you're thinking about what is really next, what is immediately next on the horizon from a gene therapy standpoint. So inborn errors of metabolism, just briefly, 1 in 1,200 people. So it's a few million people worldwide have one of these problems. And it's a big deficit in medicine because we really don't have a treatment. No one has ever been cured of their disease outside of an organ transplantation. So pretty dramatic. And then, as we know, just in this country, only about 18 to 20% of people ever make it to organ transplantation if you need one. The rest of the people don't get an organ transplant and die on the transplant list. That's a pretty dramatic problem when you're talking about the liver. So we chose to use this disease because it was a model disease to accomplish two big things. To be able to grow unlimited quantities of human hepatocytes, to treat generically lots of disorders, and you'll see how this plays in a role here in just a moment. But it also was the model disease for us to begin to treat inborn errors of metabolism and think about a solution that wasn't going to treat one rare disease, but to be able to be a platform to treat many rare diseases. And I'll try to prove that to you here in just a few slides. So we first had to create this. So we, about a decade ago, we did this work before gene editing, before all that fancy molecular technology that exists today, which makes this a lot more rapid. And we had to do this by creating an animal model that had the exact same, a large animal model that had the exact same human disease as the humans do, and prove it. And this would be our first preclinical model for our gene therapy, but also our bioreactor for growing unlimited quantities of human hepatocytes. So to make this, we had to create a virus that didn't exist. It's not naturally occurring in nature. We didn't just augment it, we literally had to create it. In collaboration with our collaborators at Stanford, we came up with a virus that was really efficient to do this AAV DJ. And this really creates this idea that you can create the virus that's a pseudotype virus. It's a blend of many different attributes of the viruses that exist in nature, plus this knockout construct that allows you to, at a molecular level, go in straight to the genome of the pig, right? The pig is great because the pig is about the same size as a human. It has similar physiology, similar immunology, what mice and rats and other preclinical models don't have. And we can make these viruses. And these viruses are smart because in the naturally occurring viruses don't have the same properties as this virus. Highly efficient, being able to target the exact cell type that we want, which is a fetal fibroblast here. And this virus has the ability to not only take your knockout construct, because you're going to knock out a gene, because you want it to behave like the human disease, but it has the ability to do so incredibly efficient. And when it does this, it does it at the genomic level. It delivers this knockout construct, and it finds its place in the genome by using these flanking homology arms. And when doing so, these flanking homology arms finds the exact place in the genome that we're looking for. So 3 billion base pairs, and we find the handful that we're looking for. It's like finding the right 1-inch square cube between Pluto and Pluto, right? I mean, it's amazing how big the genome is, and being able to find something like that in the genome very specifically without having off-target events is really challenging to do. And then you grow up these targeted cells, and they act as the substrate for cloning. And cloning is important because if you want to create a human disease in a larger animal that mimics the human diseases, you can't just use an embryo. We're not like mice. Mice, you can do that with a large animal. You actually have to clone the animal. So the cloning step is really important. You take that genetically modified cell, and it acts as the donor. So I can take eggs from the animal, and the eggs are the immature oocytes, and I can culture them. And when I take the eggs and I culture them, I can remove the genetic material out of them. And when I do that, they still have all the other potential of an embryo or an egg, right? And then I can take that cell that I created, that fetal fibroblast, and I can fuse it with that empty egg. And when I do that, I can create an embryo. And from that, the entire animal will have, every cell in its body will have that exact same gene defect. And that's how this first animal was created. And this was at a time when no one knew how to do all the steps that we just said. Now this is no big deal. To give this talk to a science community, like, yeah, they fall asleep. It's not that big of a deal. But this was just 10 years ago. So then we get these cute little guys. And this guy was the runt of the litter, and he had to wait till all of his siblings were done feeding, and he couldn't reach mom until they were sleeping, sacked out, and then he could reach. So this is the resourcefulness that we all need to have. Just to prove to you guys, and this will be the exciting part coming up, that we were able to create the human disease. Tyrosinemia is marked by grossly elevated bilirubin levels, tyrosine levels, succinate acinone levels, and a failing liver with elevated ammonia levels. And then the fibrotic disease, this is severe fibrosis that exists. These patients all develop fibrosis, cirrhosis, and hepatocellular carcinoma, and either die from acute liver failure in the first 6 months of life or die very briefly after that from hepatocellular carcinoma, just like humans. And our animals behave just the same way. At 6 months, all of them are dead, untreated. So then what we did is we then took our animal and we said, let's take our gene therapy and let's see if it works. So we had this whole idea, but we had to spend all this time creating the model that the FDA requires us to use to be able to prove that this is not only going to be effective, but safe in humans. So we make our tyrosinemia pig, and they're cute when they're piglets. They're not so cute when they're older. And this is one approach where you can do this ex vivo. Your hepatocytes are the amazing substrate for this. You can either in vivo just deliver your gene therapy construct straight to the hepatocytes, or take the hepatocytes out through a biopsy and use those hepatocytes as the substrate for your gene therapy. In this particular study, we used 4 animals that all had that human disease, that tyrosinemia, and then we took those hepatocytes and we isolated them. And when those hepatocytes are isolated, they basically, whether you isolate them ex vivo or if you isolate them in vivo through just a portal vein delivery of the vector construct, in this particular case we're using Linty, which delivers a working copy of the gene that's defective from the, to the patient. You can deliver that specifically to the cell type that you want, and then that hepatocyte ends up being the substrate for which you can then either deliver back to the patient, or because you did it through a limited approach to the portal vein, you're able to limit the expression or the distribution of the vector just to the liver. And this is what it looks like after you're done. It's a little slow here, sorry. So you can either infuse the vector or the cells back in through the portal vein, limit the distribution just to the liver, and we've done the studies that show that you can do that, and the liver corrects itself. Now here's what's special. This is why we chose tyrosinemia. This is the only one of the diseases out of these hundreds and hundreds of inborn errors of metabolism in the liver that a corrected copy, right? It's the only one that does this, a corrected copy of the cell next to if you say 0.1%, so tiny, right? If you fix 0.1% of any disease cell population, you're really probably not going to affect the disease. But in this disease, it's unique. 0.1% will become 100% every single time because of the Darwinian survival advantage of the corrected cell over the uncorrected cell. The uncorrected cell is so diseased and dies so quickly outside of therapy, and the liver is amazing. You can talk to all the physicians and doctors in the room. If I take away 80% of your liver, you're going to have, by liver mass volume, 85 to 90% of that liver mass volume back within 12 weeks. The liver has an amazing regenerative capacity. So the disease acts as a selector. It's going to kill off all the cells that aren't corrected, and only the cells that you introduced or the cells that you fixed will be the only cells that you can grow and expand. So just your corrected cells. And I'll prove it to you here. So this is what it looks like at 12 months. And this is, we've published this data over and over in multiple venues when we do in utero, in vivo, or ex vivo gene therapy. But this is a pig that weighs 130 kilos, may need some bariatric surgery. And this liver is 1.5 kilos, right? About 4 times the size of a human liver in weight. And you can see prior to treatment, this is a biopsy from this animal, no resty red-brown cells representing the absence of that functional gene, all right, that functional protein. 12 months after therapy, every part of this liver, this 1.5 kilo liver, is completely repopulated with FAH-positive resty red-brown cells. Now think about it for a moment. This is a bioreactor, right? If you've got PKU, you don't have tyrosemia. If you've got PKU, another popular inborn error of metabolism liver disease, I can take a little sample of your cells out, fix PKU, deliver the gene that you don't have, correct it or deliver it, put it in our bioreactor, grow up unlimited quantities of your cells in our bioreactor and give you back your own cells. And now you don't need a transplant. Now we're just going into the White House, changing the knob rather than knocking down the whole building to correct the problem. So we talk about this in summary. I think this is really important. This work not only I think is going to impact patients, I think there's a growing body of literature and growing interest both commercially and in academia that says this is the future. And you talk about the changes that are going to come down the pipeline. I don't think we're going to be treating disorders. I think we're going to cure them, and I think this is a big part of what that looks like. We published this particular bit of data that I showed you. We published this in Science and Translation Medicine, and the editor wrote, and I like this, editor, a member of the National Academy of Science, having demonstrated in large animals the use of the material that are safe for use in people, the technology is now poised to move into patients to regenerate their own livers and spare them the long wait times on the liver transplant list. So we'll do the surgery at 24 weeks, and then we're going to want to keep the pregnancy going, but things are going to be okay. You've got a healthy baby. We've got a problem that we're going to have to address, but your baby is going to be well cared for here.
There's your baby's hand right there. See those little fingers wiggling? Yes. Giving us a nice little stretch. Nice job, little baby.
The human body is a truly remarkable thing. At just 7 weeks, billions of cells have formed to create a baby with a beating heart. All of this is directed by genes found in the DNA, which is the blueprint for how cells work in each organ. Take, for instance, the liver. The liver is responsible for metabolism functions of the body, filtering chemicals in the blood, converting fats into energy, and breaking down toxins. It's constantly working miracles for us, and all that amazing work relies on a precise blueprint. However, sometimes the DNA has mistakes that can leave a gene completely disabled. Disabled genes can prevent proper cell function. Cells that don't function properly because of bad genes can have a range of consequences from the very minor to the life-threatening. This is where many diseases come from. 1 in 1,200 children are born with one of these diseases affecting the liver. As a whole, these diseases are called inborn errors of metabolism of the liver. 90% of children diagnosed with this problem won't live to adulthood. One such disorder is tyrosinemia, which prevents the liver from breaking down an essential amino acid found in our food called tyrosine. The result is tissue death and scarring in the liver, called cirrhosis. Now, these patients require a complex and rigorous lifelong care plan that is extraordinarily difficult to adhere to. But that is about to change.
So I'm one of the fetal interventionalists here at Children's Minnesota, and we specialize in performing surgeries on babies while they're still in the womb. There's this whole group of diseases, they're called single gene disorders. And up until now, we've only had the ability to treat the side effects of these gene disorders, but not anymore. We're part of a team that's changing that. We now have the ability to cure the disease by reintroducing the gene product that's missing. It's called gene therapy.
If we can add the correct gene to the liver cells of an infant with tyrosinemia using specially engineered viruses, we'll stop the disease before it has a chance to cause any damage. These viruses are programmed to deliver a working copy of the gene to the cells that don't have it. We can target the specific liver cells that need repair, drop the package inside, and reprogram them by adding a healthy gene, which then takes over for the inactive one. When the repaired cells replicate, they replace the old flawed cells, effectively eliminating the potential for life-threatening liver disease before any damage is caused.
Gene therapy for the liver is just the beginning. We see a future where many of these genetic disorders will be treated before our children are even born, giving them a chance at a full and healthy life. That's it.